Mitosis is the actual division of chromosomes. Chromosomes are made up of chromatin, which is thread-like strands of DNA which are condensed into chromosomes. These chromosomes are split into two daughter cells with completely new and fresh nuclei and organelles. This phase occurs for reproduction and growth, death of a cell, and loss or damaged cells. This ordinary cell division has two complete phases. The first, which is interphase. Interphase includes G1, where growth and metabolic function take place. S, which is synthesis, where the DNA is replicated. And last, G2, which is where most growth occurs and organelles are replaced. Cells spend 90% of their life in interphase. Interphase is not a part of mitosis.
Go.
This is what a cell looks like during interphase. This is the nuclear envelope. And inside the nuclear envelope is the chromatin and the nucleoli. This is the plasma membrane. These are centrioles, which are surrounded by asters, which later aid in mitosis. The three stages, G1, S, and G2, all take place when the cell looks like this.
Prophase is the first part of actual mitosis, and in this phase has an early stage and a late stage. And in this phase, the chromatin condenses to form chromosomes. It's right here. And the nucleolus disappears, and the centrosomes divide. The nuclear envelope begins to disintegrate. The mitotic spindle fibers, they begin to form and connect to the centrioles. In late prophase, the spindle fibers are attached to the centrosomes, and the nuclear envelope begins to break away, and pieces of it form around. But then here we have the spindle fibers attached also to the centromeres of chromosomes at the kinetochores, and they start pulling. Enter second phase, and then you enter into the second phase, which is the second stage of mitosis is known as metaphase. Meta meaning middle. Here, the spindle fibers tug on the chromosomes until they align themselves in the middle of the cell. The two top and bottom parts of the butterfly are known as sister chromatids.
Next is anaphase, which means opposites. This is where the spindle fibers will pull apart the chromosomes, forming two daughter chromosomes, and they will elongate the cell, making two identical sets of DNA. Prophase is the last part, and here prophase is reversed. The nuclear envelope begins to reform. The nucleolus reforms, which I'm going to show this. The chromosomes unwind and unravel back into chromatin, and the spindle fibers retract and disappear. Okay, and then cytokinesis, which is not a part of mitotic phase, completely closes the cell division process. This is where the two daughter cells become official, and animal cells form a cleavage furrow, and plant cells will form a cell plate.